Can we get rid of those things, or they're just on the screen? Okay, I'd like to start off by thanking Srikanth and the members of the organizing committee for giving me this chance to present some of my work here. What I really want to talk about is how you pattern living aggregates. And I'm not really going to be talking about either nucleation or aggregation more than as much as you see here. So I will use the verb in probably not the right context, but I'll try and tell you how one can nucleate patterns in aggregates made of living cells, and how you can kind of grow these patterns. And my focus will be on the importance of interfaces between cells in patterning these aggregates. So I will start by thanking the people who've actually contributed to all of the work that I'm going to speak to you about. And I'd like to acknowledge a talented bunch of 6 graduate students who've contributed to some or most of the work that I'm going to speak about. But I'll also briefly talk about work that's been a really rewarding collaboration with G.V. Shivshankar and Madan Rao, who's here, and I hope he'll answer any questions there are from the physics side of things, and I look forward to many other collaborations to come. So I'll start off by saying one can think of tissues as living aggregates. Madan will probably like to call cells active, deformable particles that are compacted. So, but this, that kind of doesn't describe the diversity. Doesn't describe the diversity you see in the way these cells can be packed in tissues. Some of these arrangements look seemingly simple and straightforward, but others look very complex. And all of what I've shown you here are really static time snaps or snapshots of the way these tissues look at a given point in time. But in fact, there's an added complexity, and I'll come to that in a little while. This diversity in the way cells are compacted into tissues is important because in many instances, the form of a tissue is known to directly influence its function. So just to go back to those two seemingly simple examples that I showed in my previous slide, you can still see that there are differences in the way cells are packed between these two tissues. Here you have a more ordered hexagonal array of cells, whereas here you have something that looks like a hexagonal array, but somehow it doesn't look quite as ordered. And we really want to understand how tissues can go about having these various forms. And this, again, is a two-dimensional view of what these tissues look like. But in 3 dimensions, this tissue would look something like this, and this tissue would look, it's from the wing of a fly. I'll come to that in a little while. And it's a flat bilayer. So if you actually cut them to visualize the 3rd dimension, this tissue would look like this. What you're seeing here is this bit here, right? And this tissue would look like 2 layers opposed to each other. And so it becomes important to look at tissues not just in the way they're packed in a 2-dimensional plane, but also how they're arranged in 3 dimensions. And this is where the nature of the interfaces that hold them together also becomes important. I guess the additional complexity is that these are time points. But in reality, these tissues are actually, or these cells within tissues are very dynamic, and you can see that here. So what you saw earlier on was just a static picture, and this is more like the dynamic picture. And within this, you can already see that although there's some similarities in what most cells are doing, there are also some differences, and I'll come to that in a little while. And it's not just that the cells are dynamic, but the molecules that make them up are also dynamic. And this just shows you one molecule and I'll show you these pictures in higher resolution, whose movement within these cells is also very dynamic. And so there are really two big issues here. One of how you generate diversity in the way cells aggregate to form tissues, and the other problem is one of how you integrate information through all these scales, from molecules to tissues. And one of the favorite model systems in which we begin to address these questions are tissues from the embryo of the fruit fly, embryo and adult structures of the fruit fly. And so really what we would like to ask is, where is the information for patterning these aggregates? And some of the things that we would like to be able to answer, somewhat bigger questions we'd like to answer, is how much of this is hardwired in the genetic information, and how much of it actually just emerges. And we'd really like to do this as a means of understanding how you generate diversity of form and function in tissues. We'd also like to understand this from the point of view of tissue reorganization, because these living aggregates have the remarkable ability to withstand a large number of perturbations to their integrity, either natural or unnatural, and hold themselves together. And really what we'd like to understand is what the basis of that is. I think another question that we would like to really get answers to is how tissues, or how molecules within tissues, play out the rules of physics or violate them, as the case may be, what allows them to do that. And to put this all in a more human context, understanding the way cells are packed and the way cells behave hopefully will help us one day understand how cancer spread and wounds heal. So coming back to aggregation, which is kind of the theme of this meeting, possibly one of the greatest moments in evolution was the evolution of multicellularity. And although most single-celled organisms appear to be able to live life quite happily and do most of the things they need to do, really the fact that cells could now organize themselves into collectives has led to a remarkable diversity of form and function. And much of this was really enabled by the evolution or appearance, whatever you might want to call it, of molecules that mediate cell-cell communication and cell adhesion. And so what I'd like to do now is just take you through the logic of what these molecules are that hold cells together and form aggregates. So there are a class of molecules that hold cells together, and these are called cell adhesion molecules. And regardless of what kind they are, they have a conserved logic about them, which is that part of the molecule, so pictured in this cartoon here, so in this gray area is a single cell. This is one end of the cell, this is the other end. Just assume for now that this is the top end and this is the bottom end of the cell. This is the surface on which the cell or the sheet sits, right? And these are the links that hold neighboring cells together. So there are a wide variety of molecules that hold cells either to each other or to the substrate on which they rest. All of them have one or two common features. One is that they form molecular bridges with similar regions that jut out of the membranes of the cell from the neighbors. So these bits that lie outside the cell make molecular bridges with the neighboring cells. On the inside, these molecules can, again, I'll use the word nucleate without necessarily implying nucleate the way you people do, can nucleate organization of the cytoskeleton, be it actin or microtubules. In addition to that, they can transduce signals that go into the nucleus and alter the genetic composition of the cell. And there are many different classes of adhesion molecules that are located in regions of the cells that can be seen as distinct structures if you look at it on using an electron microscope. So here's one, here's another, here's another. So they are also ultrastructurally distinct structures, and they contain these molecular complexes. And so what these cytoskeletal elements allow the cell to do is really to alter their mechanical properties. So they allow them to do things like contract, change shape, or essentially deform themselves by applying either compressive or tensile loads on the cell. And so adhesion molecules are sort of nicely located to allow the communication of not just chemical signals, but also physical forces between cells and their substrates. But having sort of spoken of them as things that hold cells together, I want to emphasize the fact that they're not glues in the sense that they stick things to each other irreversibly, but really one should think of them as dynamic seals and soles. And if you kind of visualize the way cells in tissues behave in real time, as you see here, to allow cells to do all the juggling that you see them do, you need these contacts not to be always too strong and stable. And you can also see that they need to be modulated. So for instance, somewhere here you see something different happening from somewhere here. And so therefore, it's likely that within the same tissue, the nature, the qualitative or quantitative nature of these seals and soles is also modulated to allow different kinds of behavior. So if I just take the movie I showed you a little while ago, and you may not have been very observant, but if you look at it, there are many different scales of dynamics you can visualize in this tissue. The first was what was, I think, obvious to almost everybody, which is that you had an ellipse that shrank as a function of time and was sort of reduced to a sort of straight line, so to speak. But within the cells of that ellipse, or within the cells, or in the cells that are bound within that ellipse, you can see different kinds of behavior. So this cell here in orange does this as a function of time. This cell here in green does this as a function of time. And basically the difference between this and this is that this is more or less isotropic in the way that it behaves, whereas this is a little anisotropic in the way it behaves. That's not all. We'll come to that in a little while. And then there's this dynamics here where you see that there's one central cell that behaves differently from its set of nearest neighbors. But these nearest neighbors also behave differently from the more distant neighbors. So if one just looks closely into a tissue, one can see that the dynamics of cells that make up these aggregates is actually quite complex and heterogeneous. And so really, part of what we would like to ask is how is this heterogeneity in spatiotemporal dynamics pattern? And how is it actually accommodated in tissues to still allow something more global like that ellipse shrinkage to happen pretty much stereotypically? And so I'd like to say here that, I'm a biologist and everybody in my lab does biology. So we have a set of tools that we use, that some of which you've heard from other people before, but maybe not all of them. So what we can do with these aggregates is actually perturb them in selective ways. So we can perturb them in patches, and some of these are also things that people working with colloids are trying to do. So we can perturb them in patches. So we can make clusters of cells that are different from in the tissue. We can make a specific set of cells different from other cells in the tissue. We can make single cells within the tissue different from other cells. And then we can use more sophisticated non-genetic methods to actually ablate structures on the nanoscale using lasers that can be targeted to a diffraction-limited spot. And like you also heard the people working with colloids say, we use real-time confocal microscopy to visualize dynamics of membranes, molecules within them in real time. And all of these movies that I'll show over the course of this talk are really movies that are three-dimensional over time. So each of these, at each time point, what you're seeing is a two-dimensional projection of a three-dimensional image. So it does capture all of the information in depth. So we can visualize the dynamics of the membranes. We can visualize dynamics of molecules inside these cells. And at high resolution, you can begin to see movements and streams, and I'll show them to you. And we can combine many of these things together. We can combine genetics, we can combine lasers, we can combine confocal microscopy, and then visualize the effects of multiple kinds of perturbations. Given that we can visualize all of these things, we can then go extract outlines and track particle movements and quantitatively and subject them to some kind of quantitative analysis. And then we can compare these quantities in perturbed versus native cells and draw conclusions from that. And finally, what we'd like to do, having assimilated all of this information, is really to come up with models that explain how, say, interfaces impact on cell behavior, and then work very closely with physicists to help us understand other features. So this is essentially what I've told you. What I will not be talking about today is some of the studies of us trying to look at visualize molecular interactions inside the cell in real time. Nor will I tell you about what we're trying to do with cells outside their native context. But today what I'd just like to do is focus on just this one problem, which is to look at this ellipse and ask, how does this ellipse shrink? And I won't deal with why it shrinks asymmetrically today. Try and address what drives the collective dynamics of cells at the boundary, and try and address what drives the seemingly stochastic dynamics of cell loss, and then try and arrive at some speculations of why they coexist. So one of the things, if you want to shrink an ellipse, well, essentially you're reducing its area as a function of time. We've reduced it to a two-dimensional problem, although it's a three-dimensional problem that requires you to flatten the dome and shrink the ellipse. And so what we can do is follow the areas of single cells within this ellipse as a function of time. And if we follow the area of these single cells as a function of time, you'll see that they become smaller, right? That's what this green line shows. Now, cells have been known to be able to constrict just one part, called the apical part for the top end, as a way by which they can bend a whole sheet of cells. So constricting one part of the cell to bend a sheet is commonly fairly well studied, and you can see that it does accomplish the bending of the sheet. But what we're seeing here, what we're looking at here is not really a bending of a sheet, but actually just contracting the tissue. And part of what we want to ask is, what does it take? What does it take to contract the tissue, and how do cells constrict to achieve that? And so this process of this green cell becoming smaller as a function of time is actually a complex trajectory that can be divided into a couple of phases that look like this. And what you're looking at here, it changes in area, but we've looked at a bunch of other things that I won't go into. And you can see that there is some pattern to it, even though it looks from the movies as if they're just becoming smaller as a function of time. And if you actually try and ask yourself, why is it that these areas are going up and down like this, rather than just going on a downward slope? one can get a clue to that from the behavior of some of the elements that I call cytoskeletal elements inside the cell. And so what you see here are a set of images spaced apart in time. And you can see that in some images you have these big blobs, big black blobs, and in others you don't have them. And by and large you can say that when you have this big blob somewhere in the cell looks more contracted than when you don't. And so if you actually measure the intensity of these blobs in relation to these areas, you can see a sort of negative correlation. So when the blob is there, the area is smaller. When the blob's not there, the area is larger. And this movie sort of shows you the movement of these blobs. And you can actually see that where the blob, you can actually see that it's a bit like a game of squash. When when this ball hits a wall, then it does something. And then when it bounces back, that thing kind of relaxes. So essentially, I'm going to take out all biological jargon and just say that some kinds of molecular aggregates form within these cells that appear to assemble and break up with a sort of periodicity that is consistent with the periodicity of the fluctuations. And so we can ask whether these aggregations break up in make and break of aggregations is actually meaningful. And again, we can do that by trying to perturb things that we intuitively feel might perturb the formation of these aggregates. And what you can see is so we can perturb in single cells, like I showed you before. We can take away something that destroys those blobs and show that the cell actually fails to constrict, but it actually expands. And this is the trajectory there. It's not quite, so these are trajectories of their nearest neighbors. So they're doing what? So this is the average population of nearest and distant neighbors. You see those continue to constrict in those two phases, whereas the cell that's been perturbed and in which those squash balls don't keep hitting the wall, the cells expand. But you can do just the opposite in a cell, which is to make this regulator always be on. And what you see is that the cells, again, lose their phasic behavior, but they're now sort of constitutively constricted. And this is a neighbor here that continues to behave just the same way. So basically what this allows us to say is that you can abolish these fluctuations either by having something constitutively on or constitutively off, but having it on or off affects the areas or ability of these cells to contract in differential ways. So in some sense, what we can say is that distinct kinds of aggregates or cytoskeletal elements must, on the one hand, facilitate this fluctuation behavior, and on the other hand, allow the cell to constrict. And we think that these are elements that may be, in one case, on the inside of the cell, and in the other case, along what we would call the surface of the cell. But what we can also show is that these regulators that perturb these fluctuations also perturb the way in which cells are held together. So here, what you're looking at is the distribution of one of those adhesion molecules that was on that cartoon. Forget about what it is. But in an unperturbed state, this is the pattern that you would expect to see. But when in the scenario in which you've made this perturbation, this is what happens to the distribution of adhesion molecules. So simplest way one can think about what we are seeing is that a chemical switch does something to adhesion, which then influences the organization of these cytoskeletal aggregates that then lead to pulsed cell behavior. Precisely how adhesion is functionally altered by these switches, or what the composition of these aggregates is, or precisely how their formation and disaggregation is affected is something that we're just beginning to get answers to. So now, having told you about this behavior of a cell that's all it's trying to do is constrict, I want to talk a little bit about the cells at the boundary, so cells that are at the edge of the ellipse. And there I just want to go back to some topological considerations and say that so what we've done here now is cut that ellipse or cut the embryo as if it were a loaf of bread. So the boundary of the ellipse is here, and the cells that we were looking at are these cells here. And what I'd like to point out is that the boundary, the cells at the boundary actually have distinct interfaces. Forget about what's in these interfaces. But the interface that this cell at the boundary makes with the cells on the other side of the boundary is very different from the interface it makes with cells on the inner side. So essentially, this cell poses hybrid interfaces. And if you compare the dynamics, now we're comparing the dynamics of these orange cells with the green cells. What you can say is that, so if you just look at the behavior of the orange cells, these are multiple tracks of multiple orange cells. This is how they behave. If you look at the green cells, this is how they behave. And one obvious conclusion one can make from this is that there's something synchronous about the way these cells behave. But these cells are a little less synchronous. They reach a particular defined endpoint at different times, whereas all of these reach that endpoint at the same time. So there's something collective and synchronous about these cells, and that's sort of averaged out here. There are also other differences in their precise dynamics that I don't want to go into. But so, apart from the fact that there is a hybrid adhesive interface that these cells have, there are also known gradients of chemicals that are high on one side and low on the other side. And so one of the things we wanted to ask was whether these gradients actually influence the behavior of these cells. And so what you can see here is, so what we've done here is just labeled these cells, some of these cells at the interface. And this is a native scenario. And here we've perturbed that gradient so that instead of just being restricted to this side of the interface, we've now extended it a little bit on the other side. And what you can see from these two movies, and they'll just play over time, is that these cells, whether they're white, filled white or plain white, continue to behave as you would expect these cells to normally behave. Whereas these cells here fail to become smaller over time, but instead actually become larger. Right? So they appear to elongate. So their areas go up like this, and then catastrophe happens, and they don't finish what they're supposed to do. So what one can conclude from that is that just playing around with a chemical gradient can deform a cell so that it now fails to contract, but instead starts to elongate. And then you start taking a look at how adhesion molecules are distributed at these interfaces. You will see that in a native, unperturbed cell, this is what adhesion molecules look like in this row of cells. Whereas if you either completely abolish any low level of this gradient that might have been in this periphery, or you exaggerate that gradient on the other side, you see opposite effects on the distribution of adhesion molecules. In one case, it looks as if you're forming new clumps. In the other case, it looks as if you're breaking the continuity of the distribution. And so one can quantify this in different ways and show that in both these cases it's significant. So what one can conclude from this is that you can have a chemical gradient can influence or is sufficient to deform cell behavior, but it appears to do this through its influence on the adhesion complex, and possibly through its influence on the mechanical properties of these cells. And again, the kinds of questions for which we've just begun to get answers is, what do these clumps and breaks in adhesion molecule distribution actually mean? What does that mean in molecular terms, and what does that mean in functional terms? And I'll take you through some assays that we think may help us understand this. Right. And so one of those assays that actually helped us understand a little bit about looking at adhesion in quantitative terms is really something that we set out to do in collaboration with Shiva at NCBS. And what we set out to do was really to try and see if we could induce patterns in tissues by ablating structures, although that wasn't quite why we set out to do this. But in any case, so what we can do is use lasers targeted to a diffraction limited spot to ablate any structure within the cell or in the membrane, and then look at how that influences behavior. What you'll see if you do such a thing is that the cell expands first, right? This is the cell that I was just showing you. It expands first, and then it contracts and creates this rosette-like structure that you may remember from my first slide. So if you look at it in time, this is how the pattern evolves. Again, one can watch how the area changes as a function of time, and you see that it increases and then falls. And its nearest neighbors here don't show much change, although they're very clearly deformed in shape, right? And you can quantify that anisotropy in shape by measuring the aspect ratios of the cells as a function of time, and you can see that it changes sign during the course of this. And with the help of Madan and the theory that he put together, one can actually begin to use some of these quantities to measure, to get a sense of the physical constraints that hold these cells together in these tissues in the first place. So one can argue that, the velocities, the recoil velocities of these membranes, when you subject them to this kind of ablation, must tell you something about the physical constraints that hold cells together. So hold that thought for a minute, and I'll come back to this in a little while. So now that you can induce a pattern that's not very different from one of the patterns that I showed you in the native state, apart from the fact that there's this intermediate state of expansion, one can begin to ask, how does a cell actually create this pattern, right? Because all that was done was actually make a small nanoscale perturbation inside the cell. And so really what we wanted to ask is, how does an aggregate arrange itself to form a rosette? And I told you earlier on that the chief elements that allow a cell to change shape are the cytoskeletal elements. So we started by looking for rearrangements in these cytoskeletal elements upon this ablation. And so one of the elements that we looked at shows this. So this dot here marks the cell that was perturbed. And this element here begins to appear in the form of ruffles in a ring, right? And these ruffles kind of move inwards, and I'll show you a movie of that. And then subsequently, there's a sort of enrichment around the membrane before the cell becomes really small. Another cytoskeletal element starts to show something that looks like flows that are directed towards this interface, that subsequently lead to a very tight ring as the cell contracts. So if I just had to show you what each of these elements looks like, this is the one that I showed you on top. So you can see these things going inwards, right? And I should just point out that all of these structures are actually coming from the neighbors and not from the cell itself, and there are ways we've worked this out. And then only later on you see something that looks like a ring that's forming and that contracts this cell, right? If you look at this other element, the dynamics of it are quite different. So you can see that there's a continuous streaming of particles towards that cell that expands first and then contracts. But again, there you see a ring forming in the end. So, but there's a flow towards that interface that precedes the formation of that ring. And then there are other kinds of elements one can look at that seem to form largely structures that look like asters. And again, here's what you see. So you can see some sort of lines here that are all pointed towards this new interface. And these are somewhat harder to visualize in movies because they're very fine structures, but you can sort of begin to get a sense of them for polarizing their alignment towards this interface. So basically, through a combination of studies, we can sort of say that there's a sequence in which these rearrangements are deployed to allow this pattern to evolve. And this sequence includes ruffles, it includes rings, it includes flows, and it includes asters, all of which happen in a particular, deployed in a sequential kind of way. And so one can ask whether any of these things are meaningful, so you can break up these asters using genetic tools that break up the elements that make those asters, and ask, now when you make those ablations in them, do they actually make good rosettes? And you can look at this in different ways. You can ask, how perfect is it? Do all cells show those anisotropies, and all the neighbors show anisotropies in shape? Do they expand to the same extent as their neighbors, and do they contract to the same extent? And you can see that the orange line here represents the cells that have been perturbed, whose asters have been broken, so to speak. And this is the control. And there is some difference in the way this pattern evolves, both qualitatively and quantitatively. You can also ask, if you change the properties of the interface in these cells, what happens? And again, what you can see is you can do it in many different ways. And if this is the way in which the area evolves as a function of time, one of two different kinds of perturbations in adhesion also alters the way that this response happens. But you can also show that altering the interface actually prevents the, sort of crystallization of this polarity, so to speak. So you begin to see, in large number of cases, these ruffles are pointed all over the place. They're not going inwards into the cell, but in some cases they're also going on the other side of the cell. And the ring doesn't appear to form as crisply as it should. So clearly something about the properties of this interface are essential to generate that pattern. And this just shows you one of those perturbations in which you begin to see some things happening normally, but then things start going haywire, and you start seeing structures going all the way out, and then from a phase of expansion, you start to contract, but then you expand again, and so on. So clearly, what that suggests is that interfaces, or the composition of these interfaces, influence these emergent polarities. And I think what we don't quite understand yet is how that one interface that it makes with all its nearest neighbors is polarized. And what, if anything, accounts for the stochasticity in the same kind of pattern that happens in the native state. But one of the things that this kind of ablation allows us to do is to break cytoskeletal structures, or break elements that we think confer mechanical properties on the cell, or apply forces onto a cell, and therefore infer the influence of a mechanical stimulus on cell behavior. And we hope to continue to apply more local forces using other techniques in collaboration with Shiva. But one of the things I'd like to return to is to the interface and ask and go back to the issue of how it alters the properties of the cell. And I told you that cells at the boundary actually have hybrid interfaces. They have one kind of interface here and another kind of interface with either these neighbors or these neighbors. And if this interface was actually very different from all of these at some functional level, then one would argue that if you looked at the response of the cell to the kind of laser ablation I showed you, you would expect to see some kind of anisotropy in it. And this is what we see. So when we make an ablation here, you can see that the cell expands on all these interfaces. But this interface stays nice and taut. And so there's a quantification here that looks at aspect ratios with respect to a fixed, not just major versus minor, but you fix one of the axes and look at the dimensions of the axis perpendicular to it. And what we can also show in the situation where we altered the gradient across this interface is that the response of an unperturbed interface versus a perturbed interface are very, very different. So this kind of allows us to say that in that situation where we played around with the chemical gradient, there may be this adhesion complex, which we saw was broken into clumps, or its continuity was lost. Maybe it's altering the, it's actually changing the mechanical properties of the cell as evident from its response to ablation. And I think I'm nearly done, but I'll just spend a few minutes to say that that one can see many different kinds of patterns, suggesting that we don't completely understand all the ways in which cells can communicate across interfaces with their neighbors. And so here again, we go back to tools that we can use to perturb single cells that are shown here. You have a doublet here that both look filled up. These are perturbed. And with some kinds of perturbations, what we can do is influence a large proportion of its nearest neighbors to actually contract, like in just the same way that so I'll show you what I mean. So basically what you can do is have some cells there. So we have two patterns of behavior. In some cases, we perturb one cell and then there's a relay, one cell after the other just contracts very, very fast and goes away. In the other case, you have cells and a large proportion of its nearest neighbors actually contract and then disappear, right? So what's plotted here is just the frequency of nearest neighbors that actually exhibit a behavior in relation to those that aren't perturbed. And we've identified signals that could now allow cells in a row to behave the same way, or cells in a ring around a perturbed cell to behave the same way, and we're beginning to understand how these modes of communication work. So I think what I've told you so far is that adhesion can have, can enable very complex interactions between cells, and part of the ways by which it probably does that is by making one kind of interface distinctly different from another kind of interface. In some cases, we understand what the differences in these interfaces are. In other cases, we don't really understand it. And yet, one can find some common themes in the way the cytoskeleton reorganizes in distinct places. Even though these behaviors are different, there are some commonalities in the way in which the cytoskeleton reorganizes. And one last point that I'd like to get to, just to illustrate that, well, just to make it seem like the plot thickens, is that, I said that there are all these heterogeneous behaviors that coexist within the same tissue. And so there's some evidence that there is coordination between these various behaviors. And people have actually used lasers to drill big holes in these tissues, essentially to kill off all the cells, and shown that, you can make quite huge, quite large nicks inside this ellipse, but not really affect the rate at which the ellipse shrinks very much. It's only when you make large sweeping incisions within the ellipse, and many of them, that you actually delay the fall in area substantially. And people use this to say that there's a lot of resilience in this process. If you upset one tissue, then what's on the other side of the ellipse can regulate itself to still make the rate, still make this process continue as if nothing had happened. And the proof of this was that if you made two large nicks at the edges of this ellipse, the bit in the middle actually got close to each other faster or more quickly than the bits at the edge, right? Compared to one where no nicks were made. From a genetic perspective, what we were able to show was that it's sufficient to perturb one tissue to prevent this process from happening, right? So it's not entirely true to say that there are multiple contributors and one can upregulate the other, but what genetics has allowed us to sort of say better is really that you can perturb something in the neighboring tissue and influence cell behavior in this tissue, right? So you can do something here and influence the frequency of some behavior that happens here, right? So if we look at perturbations that either make the rate at which this ellipse contracts faster or slower than what's normal, you can show that part of the reason for this is because you're influencing cell behavior here, as indicated here by either the time taken for this ellipse to close, or if you look at the frequencies of those behaviors. But what's surprising is that you can make a perturbation in the neighboring tissue and still influence all of these behaviors here. So what that tells us is that there's a sort of feedback signal that goes from one tissue to the other that can influence cell behavior in a non-autonomous kind of way, and understanding how this tissue can actually influence something happening here, whether it's purely mechanical or mechanochemical or purely chemical, is something we're just beginning to understand. So I'll just end and summarize what I've said by saying that adhesion operating at cellular interfaces can act as both contact and chemical, but it appears to be a key mediator of both cell and cytoskeletal dynamics. And I think we've reason to believe that stresses operating on the cell through adhesion molecules can influence signaling in that cell. And signals can also work via adhesion to influence the stresses that a cell experiences. And really working out the details of this is how these stresses are sensed and transduced is something that we're quite interested in doing. So as biologists, we've really focused on understanding how information is integrated to allow heterogeneities and regulation of cell behavior. We're focused on understanding what the molecular and temporal hierarchies are that allow the evolution of patterns. And what we're kind of striving to do with physicists is really to understand how these molecules feed into the physical properties of cells and what allows these cells to violate those properties. And I will just end there and take any questions.
Showed a contractile cell aggregate.
Is that turned on?
Yeah.
Okay, in the first part, you showed a contractile cell aggregate in which the cell at the boundary behaved differently than a cell in the bulk. So can that be modeled by just saying that they're in a different environment and the surface energies are different? Because the thing in the bulk looked like a contraction of a droplet, and in the surface it looked like a spreading of a droplet. So can that be?
So you're asking if they can be if that behavior can be modeled just by
considering that they're
saying that the one parameter that's different is the adhesion at the boundary. Is that your question?
The surface energy at the cell boundaries are different.
I don't actually know if it can be modeled like that. But what I can say is that we can perturb just the boundary. We can perturb just that interface, and these are experiments in progress. With different kinds of perturbations that we think either affect the floppiness of the membrane or just simply affect adhesion without affecting floppiness. And we get somewhat different results in the two cases. So I don't actually know if it can be modeled just by taking into account surface rigidity, but that's not something we've checked. And that's Madan's. Madan will tell us.
If you perturb the cells in the middle, yeah, then it turns out that the stresses don't propagate much. If you look at the neighboring cells, only beyond the first neighbors, the area and the shapes of the cells don't change much. So the stresses don't propagate too far. However, if you perturb the cells at the edges, at the edge of the teardrop, then it seems to affect the boundary. So, so when you're in the middle of the cell, it's appropriate to think of it as a fluid. Whereas if you're at the edge there, then elastic considerations are important.
So I was wondering, continuing what Madan said, if what he says is right, then if you make local probes of mechanical rigidity, of elasticity, by putting a bead in there and wiggling it at various places, you should actually find measurably different results. So is this a practical experiment?
Yeah, so we are actually trying to inject beads of various kinds, and Roop Malik, my colleague, is kind of helping us with it.
I see. Any other questions? Okay, so let's thank Maitreyi again.